Hello everyone, welcome back to the session. So in today's class we are going to discuss about an electron transport chain. So we have started the cell biology and we have completed that, and in today's class we are going to discuss about this electron transport chain and oxidative phosphorylation. With that, the entire biochemistry topic has been over. So far we have completed 2 units successfully. Without any delay, and in the cell we have started the cellular respiration. So in the cellular respiration we are having 4 stages, that is glycolysis, pyruvate decarboxylation, Krebs cycle, electron transport chain with oxidative in the means of oxidative phosphorylation. So this is the final stage and the most important step, and with that the biochemistry topic will be completed. I will start into it. So electron transport chain. So as the name suggests, see here, electron. So electron plays an important role, and the electron is transporting and it is in the chain reaction. So that's why the name electron transport chain. So the electron transport chain and oxidative phosphorylation are the final stage of the aerobic respiration. So electron transport chain and oxidative phosphorylation. So what is meant by this electron transport chain? What is oxidative phosphorylation will be discussed in the upcoming slides. Both are the final stage of the aerobic respiration. So here, so when we are studying about the Krebs cycle and the electron transport chain, we don't speak about the anaerobic respiration. But in the glycolysis, we have studied that glycolysis in aerobic respiration and glycolysis in anaerobic respiration. But the Krebs cycle and electron transport chain will be happens only with the presence of the oxygen. So that's why we didn't speak about the anaerobic respiration. And here, what is the ultimate goal of this electron transport chain and the oxidative phosphorylation? ATP is the one going to produce. So we are going to see how it's that. So but before that, you should know where exactly this electron transport chain will be taking place. So but before that, so this is the glycolysis. So this is the cytoplasm. This is the cytoplasm, and here where the glycolysis is taking place. The glycolysis in the glucose will be converting into pyruvic acid, and this cycle is called as the glycolysis process, where the glucose will be converting into the pyruvic acid in the cytoplasm, and with the means of this, with the help of the glycolysis cycle, ATP is going to produce. Now after that, the pyruvic acid will be converting into acetyl-CoA, and the Krebs cycle will be continued, and as a result ATP will be produced. Now in the glycolysis also and in the Krebs cycle also, we are having an electron carriers, and that electron carriers, they are nothing but NADH and FADH₂. Both have been produced in the glycolysis and the Krebs cycle. So both were considered as an electron carrier. So that plays an important role in the electron transport chain. Again, it will be taking place in the mitochondrial matrix, and as a result of this electron transport chain also, we are going to produce this ATP. So what is the function of this? What is the ultimate goal of the cellular respiration? We are having 4 stages, that is glycolysis, Krebs cycle, pyruvate decarboxylation, electron transport chain. With all these 4 stages, what will be the end product? That is nothing but an ATP. So different cycle, these are all the different cycles and different reactions is going to taking place, and we have completed what are all the reactions will be taking place in the glycolysis, pyruvate decarboxylation and the Krebs cycle. Now we are going to study about the what are all the things which are happening in the electron transport chain. Now look at this. This is what an electron transport chain reaction. See here, what is this? This is what a cytoplasm. This is a mitochondrial matrix region, and this is the membrane region, and here we are having a NADH dehydrogenase complex, and this complex is cytochrome BC1 complex and cytochrome oxidase complex, and we are having ATP synthase. That is, this is the one NADH dehydrogenase complex. This complex is called as the NADH dehydrogenase complex, and this complex is called as the cytochrome BC1 complex. This complex is called as the cytochrome BC1 complex, and this complex is called as a cytochrome oxidase complex, and this is called as an ATP synthase complex. Now, in NADH dehydrogenase complex, what is the thing? See here, NADH plus H plus. As a result, they are forming an oxidized state. So, whenever you are seeing a complex called NAD plus, it is in an oxidized state. When this NAD plus is undergoing a reduction reaction, so that is NADH. This is the reduction state. Now see here, NADH, which is in a reduced state, which it is converting into an oxidized state with the help of this NADH dehydrogenase complex. Whenever they are converting from reduced reduction state to an oxidized state, here where the electron is released, electron will be released here. And what is the important molecule which is involving here? FMN, flavin mononucleotide. So whenever, see, just look at this thing, whenever this NADH plus H plus is converting into an NAD plus, an electron molecule has been released, and FMN has been, with the help of this FMN, and the two proton is getting pumped to the outside. So all these processes taking place in the complex 1, that is NADH dehydrogenase complex. Now this electron has been accepted by this Q molecule, and this Q is nothing but an ubiquinone. So here where this FADH₂, again in a reduction state, that is in a reduced state, this will be converting into an oxidized state, that is FAD plus 2H plus. FAD plus 2H plus. And this 2 plus, this is nothing but a proton. Here also a 2 proton is getting pumped to the outside region, and this electron has been accepted by the cytochrome BC1 complex. So these are all the electron release of energy, and the electron is getting carried to the carried by an electron acceptor, and it has been donated, and that released electron is getting accepted by some other electron carrier. Whenever the electron has been shifted from one carrier to another carrier, there will be the release of energy, and that energy, with the help of this ATP synthase complex, the energy is getting produced. So let me explain you little bit elaborate manner in the upcoming slides. Just as of now, know this is how the reaction will be. I'll explain the slide once again. See, this is what an intermembrane space. Intermembrane space in the sense, so in the mitochondria we are having an outer membrane and the inner membrane. The inner membrane undergoing a folding region, and this folding is nothing but a cristae. Inside the cristae, that region is called as a matrix region. The outer membrane and the inner membrane, between these two membrane we are having an intermembrane space. So the intermembrane space, in this particular membrane we are having a complex 1, complex 2, this is the complex 2, and this is complex 3, complex 4, and this is what an ATP synthase region. And this complex 1, 2, 3, 4 together, in combining with all these complexes, we are saying this particular chain as an electron transport chain. And finally, where the ATP is getting produced with the help of this ATP synthase complex, this ATP synthase is also called as an F0 F1 complex. F0 F1 complex. So this complex, they are producing an ATP, and that particular process is called as a chemiosmosis process. So this is the matrix region. This is the membrane region, inner membrane and the outer membrane. Sorry, this is the inner membrane, and this membrane, this is the intermembrane space, and here we can see the outer membrane. So in the membrane we can see 4 complexes, and all these complexes, they are nothing but they will be the involvement of enzymes. Each enzymes we are naming under the different complex. That is, complex 1 is NADH dehydrogenase complex. Complex 2, succinate dehydrogenase complex, we are going to see about that. Just know that these are the electron carriers. These complexes are not nothing but an electron carriers. Whenever they are carrying the electron, whenever this NADH is converting into NAD plus, some protons has been flow from the electron is getting flow, and the proton has been pumped from the matrix region to the intermembrane space region. So the membrane, the outer membrane and the inner membrane, between these two membrane we are having a space, and that space is called as an intermembrane intermembrane space, and whenever there is a conversion from this NADH to NAD plus, FADH2 into FAD plus. So whenever there has been a conversion like this, proton has been pumped from the matrix region to the intermembrane space. As a result, there will be more proton gradient has been seen in the intermembrane space. So how many number of proton is getting pumped from matrix to the intermembrane space by each complex? That will be your question. So from at the end of this, we are having an ATP synthase. So in the ATP synthase complex, they are pumping a proton back from the intermembrane space to the matrix region. All the proton has been pumped from the intermembrane space to the matrix region. Now ADP plus P is being converted into the ATP with the help of this ATP synthase complex. Now, so let me explain you what will be the main motive of this electron transport chain. See here, electrons from the NADH and FADH2 are transferred through a chain of the carrier. So we are having a certain carrier. So this is the electron, this will be the electron donor. So see here, this is in a reduction state, the reduced state. So here the acceptance of an electron, the gain of electrons, and whenever it is getting converting into an oxidized state, that is a NAD plus, an amount of electron is getting released. So that's what we have said, the basic difference between the oxidation reaction and the reduction reaction. So in the oxidation reaction, there will be the acceptance of oxygen, removal of hydrogen, and the removal of electrons. So this is why, so NADH is been converting into NAD plus. So whenever it is converting into an oxidized state, some amount of energy is getting released. And the release electrons have been accepted by some electron carriers. We are going to see one by one everything. See, the electrons from NADH and FADH2 are transferred through a chain of carriers. There will be more number of carriers. In each carrier, some amount of electron is getting accepted. So this transfers releases energy. Whenever there been a transfer of an electron, there has been a release of energy. Now this energy is used to pump the protons. The energy is used to pump the protons across the inner mitochondrial membrane. See here, the energy which is used to pump the proton. See here, very important point. Whenever there will be the release of electrons, there also some amount of energy is going to release. This energy is being used to pump the protons from the matrix region to the inner mitochondrial membrane region. This is very important. Let me explain you once again. See, whenever this reduced state of NADH and FADH has been converted to the oxidized state, whenever some molecule is in oxidized state, they are tend to release the electrons, and that electrons from these NADH and FADH2 has been transferred through a chain of carriers. There must be some amount of carriers. See, one carrier is donating the electrons, is releasing some electrons, and that released electrons is getting accepted by some other electron carriers. Whenever the release of electrons occurs, there might be some sort of energy is also going to release, and the released electrons is used. The energy released is used to pump the proton, that is the H plus, from the matrix region to the intermembrane space region. So as a result, there will be the proton gradient. The proton gradient will be higher in the intermembrane space, and this higher proton gradient in the intermembrane space will make the ATP synthase enzyme to pump the proton back to the matrix region. This is how the ATP is going to produce. So this is what a key idea of this electron transport chain. Now, the cell type and the location of the electron transport chain. What will be the cell type? Prokaryotes and eukaryotic region. So the eukaryotes, this will be the cell type, the eukaryotes, and the inner mitochondrial membrane. Inner mitochondrial membrane, the location of ETC, which means what? In eukaryotic region, the electron transport chain will be taking place in the inner mitochondrial membrane, and in the prokaryotes, the electron transport chain will be taking place in the plasma membrane. This is very, very, very important. This can be expected in our exam. Now see, we are having a 5 major important protein complexes, and 2 are the mobile carriers. So we are having 5 protein complexes, in the sense complex 1, 2, 3, 4, 5. 5 complexes. Where among the 5 complexes we are having 2 mobile carriers. 2 mobile carriers. This can also be expected. This is the complex 1. Complex 1, we are calling them as the NADH dehydrogenase complex. Complex 2, this is called as the succinate dehydrogenase complex. Complex 3, cytochrome BC1 complex. Complex 4, cytochrome C oxidase complex. Complex 5, ATP synthase complex. Then what about this coenzyme Q and cytochrome C? Coenzyme Q is nothing but a ubiquinone, and cytochrome C is nothing but a mobile carrier. And the complex 1, NADH dehydrogenase complex. Dehydrogenase, in the sense they are accepting an electrons from the NADH. See here, NADH is the one donating the electron. They were all the release of the electrons. They were all the donor of the electrons. They are donating the electrons, and the electron which is released from this NADH has been accepted by a complex 1, and that complex 1 is nothing but the NADH dehydrogenase complex. And the complex 2, which is succinate dehydrogenase enzyme, and this is the enzyme which is accepting the electrons which is released from the FADH₂ complex. FADH₂ is releasing the electrons. FADH₂ is the one releaser of this electrons. And that electrons has been accepted by some enzyme, and that is succinate dehydrogenase enzyme. And coenzyme Q, and this coenzyme Q is named as the ubiquinone complex, and this is the mobile electron carrier. So see here, we are having 5 major protein complexes and 2 mobile carriers. So these are all the 5 complexes, and besides these 5 complexes, we are also having 2 mobile carriers also, and that carriers are nothing but this coenzyme Q and cytochrome C. Coenzyme Q is nothing but a ubiquinone. And now comes to this complex 3, cytochrome BC1 complex. They are the one transfers the electrons to the cytochrome C. So for the electrons from the cytochrome, the complex 3 to the electrons has been shifted to the complex 4 that has been accepted by the cytochrome C. Cytochrome C, the cytochrome BC1 complex transfer the electrons to the cytochrome C. And besides, so we are having a cytochrome C. This is a mobile carrier, which means what? This is the one carrying the electron from the cytochrome BC1, and it has been donating to the cytochrome C oxidase. Now here they reduces these oxygen, and it is being converted to the water, which means they are accepting the hydrogen. They are also gaining the electrons. So this is getting reduced into water. So this is very important point. Now complex 5, ATP synthase complex. So the ATP synthase, the function of this ATP synthase is main important thing is production of ATP. Now just look at this, very very important. I'll explain you one by one clearly. See, the complex 1 is NADH dehydrogenase. NADH dehydrogenase. This is the complex 1, and here where the NADH is getting converting into NADH plus. As a result, the electrons is getting released. The release of electrons from the NADH complex is being accepted by this NADH dehydrogenase complex. Now here also complex 2, which is nothing but a succinate dehydrogenase. This is what a succinate dehydrogenase. Whenever the FADH₂ is converting into FAD plus, some amount of electron is getting released, and the released electrons is being accepted by the succinate dehydrogenase complex. Now complex 3 is nothing but an ubiquinone, which is represented as Q. Now this is considered as a mobile carrier. So this is the one carrying the electrons from the succinate dehydrogenase, and it has been donated to the cytochrome C. And now from the cytochrome C, it is also converting the, which means cytochrome C is a mobile carrier and the ubiquinone is also a mobile carrier carrying the electrons from the ubiquinone. It has been transmitted to the cytochrome C oxidase enzyme. Now this is what a complex 4, and complex 5 is the ATP synthase. So this violet color is called as an ATP synthase complex, and this is the one involving in ATP synthesis process. How they are synthesizing the ATP? See here, whenever some amount of electron is being transferred from one electron carrier to another electron carrier, some amount of energy is released, and that energy is used to pump the proton from the matrix region to the intermembrane space. As all the proton is getting pumped from the matrix region to the intermembrane space, now what happens in the intermembrane space? The proton gradient will be higher in the intermembrane space. Now again, to produce the ATP, again to produce energy, this electron, the proton has been pumped from the intermembrane space to the matrix region with the help of this ATP synthase complex. And this ATP synthase complex is also called as F0 F1 complex. So this is very, very important. And besides all these things, you should know complex 1 has how many subunits? Complex 1, 47 subunits, complex 2, 4 subunits, and complex 3, 11 subunits, complex 4, 13 subunits, complex 5, 17 subunits. So these are all the number of subunits in each complex region. So have a look at that. Now see here, let me the same diagram but in some amount of additional information. This is the cytoplasm. This is the cytoplasm region, and this is the membrane. And see here, this is the mitochondrial matrix. This is the outer membrane and the inner membrane, and between these two membrane we are having an intermembrane space. And here where we are experiencing high H plus concentration. Why? Because the proton from the matrix region is being pumped to the intermembrane space region. So higher H plus concentration results in the higher acidic environment, which means the low pH. Low pH means acidic environment. Acidic environment in the sense higher H plus. So this is what the condition or the environment seen in the intermembrane space region. Now in the matrix region we are having a higher pH and low H plus concentration, because the H plus in the matrix region will be pumped to the intermembrane space. So as a result, the H plus concentration will be lower in the matrix region. As a result, we are experiencing the low H plus concentration. If there will be low H plus concentration, then the pH will be higher. And that is a basic environment. See here, basic or alkaline environment. So this is where the complex 1, and this is the complex 2, complex 3, complex 4, and complex 5. So here the same thing. Yes, NADH has been converted to NADH, same information, and here also the Krebs cycle is being taking place in the matrix region, and the H plus will be released from the Krebs cycle is also increase the proton concentration in the matrix, and that been pumped to the intermembrane space region. Now little bit elaborate one by one, little more depth analysis. Flow of electrons, how the electrons will be flowed from one carrier to the another carrier. So right from the complex 1. So the complex 1, they are nothing but this NADH dehydrogenase complex. So the electrons been released by the NADH, and it has been accepted by the complex 1. This complex 1 is nothing but NADH dehydrogenase complex. See here, the NADH gives electrons to the complex 1. This is the first information. Now from the complex 1, it has been passed to the coenzyme Q, which is nothing but an ubiquinone. Then from the complex 1, it has been accepted by the coenzyme Q, and it is not donating to complex 3, donating to complex 2. Instead of that, they are donating to complex 3. and here the electron has been donated from 1 to 3 with the help of this coenzyme Q, that is nothing but an ubiquinone. Now from the complex 3 it has been carried by the cytochrome C. Again this is also a mobile carrier. This is a mobile carrier and this is also a mobile carrier. From the cytochrome C, it has been donating the electrons to the complex 4, which is nothing but the electron has been accepted by the cytochrome C oxidase enzyme. Now the oxygen accepts the electron, then forming a water. So you should know the final acceptor of oxygen will be the complex 1. Final acceptor of the electrons will be this oxygen. Why? Because let me show you the picture. Just look at here. The electrons from the complex 1, it has been transmitted to the complex 3 with the help of this ubiquinone. From the complex 3, the electron has been accepted by the complex 4 with the help of the cytochrome C. Now there will be no involvement of complex 2. So and from the complex 4, the electrons been accepted by the oxygen, and the oxygen, by accepting the electron and the proton, they have been converting to the water. So the question can be asked, who is the final acceptor of the oxygen in the cellular respiration? You should know that will be the oxygen. Oxygen will be the final electron acceptor. Now, what is the point of having this complex 2? Because from the complex 1, the electron been accepted by the complex 3. Now, where is the complex 2? What it is happening? The complex 2 has been the electron from the FADH₂. The electron from the FADH₂ gives the electrons to the complex 2. FADH₂ gives the electrons to the complex 2. The electrons go to the coenzyme Q. See, now who is the donor of electron to the complex 1? That is nothing but an NADH. Now who is the donor of electron to the complex 2? Then that will be the FADH₂, no doubt about that. Now from the electrons from the complex 2, again it will be taken by the coenzyme Q, again ubiquinone. Then follow the same path as the NADH. See, very important. So by complex 1 we are having two pathway. One is complex 1 pathway and complex 2 pathway. So the complex 1, nothing but the donor of electron to the complex 1 will be NADH. From the complex 1, the electron is being transferred to 3 and 4. This is how it will be. And see here, who will be helping here? Coenzyme Q, and here the coenzyme C, cytochrome C. And for the complex 2, who will be the donor? FADH₂ will be the electron donor, and it has been accepted by the complex 2. With the help of this coenzyme Q, that is ubiquinone, the pathway complex 3 has been accepting the electron from the coenzyme Q. And again the cytochrome C is accepting the electron from the complex 3, and it has been donated to the complex 4, which is nothing but a cytochrome oxidase. And finally, who is now accepting the electron? Oxygen. Oxygen, by accepting the electron and the hydrogen, they are converting into the water. Again here also the final acceptor of electron will be the oxygen. So this is what the complex 1 pathway and complex 2 pathway. Complex 1 is undergoing certain reaction, complex 2 is undergoing a certain pathway, but both are same. The donor is different. Yeah, see here, as electron move, energy is used to pump the proton. Whenever the electron is moving from one carrier to another carrier, the energy is being released. The released electron is used to pump the proton from the matrix region to the intermembrane space. So see here, complex 1, complex 2, 3 and 4. Complex 1 is used to pump the proton, and this is involving in pumping the proton. Complex 3 is involving in pumping the proton. Complex 4 is involving in pumping the proton, but not the complex 2. So this can also be expected. So whenever, because the complex 2 is carrying the donating the electron to complex 3 with the help of this coenzyme Q. Whenever the electron is transferring here, this energy is not involving in pumping the proton. This can also be expected in our exam. So this is the holistic picture. Now if you see the picture, you can easily understand. For the first few slides you won't get anything, but in this slide you can get some idea. See here, this is an intermembrane space, and this is a matrix region, and this is the complex 1, and this is the complex 2, and this is the complex 3, and this is the complex 4, isn't it? See here, in the complex 1, this NADH, so from the citric acid cycle, we have studied 3 per turn 3 NADH will be produced, and 1 NADH will be involving in donating the proton, donating the electron to the complex 1. See here, the NADH, whenever it is reduced, whenever it is in an oxidized state, the oxidized state means what? They are releasing a proton, they are releasing an electron, and they are releasing an electron here, and that electron been accepted by the complex 1. And from the complex 1, it has been, the electron has been carried by this coenzyme Q, which is a mobile carrier, and as it is moving, we are saying that as a mobile carrier. From the coenzyme Q, it has been the electron will be donated to the complex 3, and from the complex 3, the cytochrome C is accepting the electron, and it has been donating to the complex 4. This is how the complex 1 pathway will be. And the second complex, that is the succinate dehydrogenase complex. The succinate molecule is also from the citric acid cycle. The succinate, that is what the succinate dehydrogenase enzyme. See, that is what a complex 2. And here, whenever the succinate is converting into a fumarate, FADH₂ will be produced. We have studied in the Krebs cycle that FADH₂ is also converting into an oxidized state. Whenever any molecule is converting into an oxidized state, they are releasing a proton, they are releasing an electron. The electron will be moved from the electron will be, now they are donating the electron, and that has been carried by the coenzyme Q again. Now this coenzyme Q is donating the electron to the complex 3, and from the complex 3 it has been donated to the complex 4. So the complex 1 is called as the NADH-COQ oxidoreductase. Complex 2 is called as the succinate dehydrogenase. Complex 3 is cytochrome BC1 complex. Complex 4 is cytochrome C oxidase. Okay. Now, so in both of these complex, that is the complex 1 pathway and complex 2 pathway. For complex 1, who is the donor of electron? NADH. For complex 2, who will be the donor of electron? That is FADH₂. So in the complex 1 pathway and in the complex 2 pathway, in both pathway, who will be the final acceptor of electron? That will be the oxygen. By accepting this electron and proton, they are converting into water. So this is the holistic view of the electron transport chain. Now what will be the result? So we have studied the proton which is present in the matrix region will be pumped into the intermembrane space. As a result of this pumping, the proton gradient will be higher in the intermembrane space and it is lower in the matrix region, and that's why we are saying that here will be the low pH and here it will be the higher pH, because of the H plus concentration in the membrane space and the matrix region, and as this forms a proton gradient here. Now see, this is what an little bit elaborate. See, some more additional important is additional point has also been added here. See, electron transport chain with the chemiosmosis. See here, electron flow. The electron has been flowed, and in addition to, whenever the electron is flowing, energy is also released. So electron flow and energy release. Now the proton movement and gradient formation. Proton movement and gradient formation in the sense, the proton is from the matrix region, it has been transported to the membrane space, and as a result of higher concentration of H plus in the intermembrane space and lower H plus concentration in the matrix region, there has been a proton gradient formation. So now this proton motive force, that is what this PMF, driven ATP synthesis. Now the proton again from the intermembrane space is pumped to the pumped to the matrix region with the help of this ATP synthase complex. In ATP synthase, we are having a 2 subunit, and that is nothing but this F0 and F1 complex. For the synthesis of ATP, how this proton gradient will be helpful, we are going to see about that. See, again, complex 1, we know that that will be the NADH dehydrogenase, complex 2 will be the succinate dehydrogenase, complex 3 will be the cytochrome BC1, and complex 4 will be the cytochrome C oxidase. This is how it will be. And see, look at this picture. So complex NADH and FADH₂. So this can also be expected in our exam. So this is what a complex 1. NADH is a complex 1 donating an electron to complex 3 and donating the electron to complex 4. That's all about the complex 1 pathway. In NADH₂, complex 2 will be the acceptor of electron from the FADH₂. So that's why complex 2, it is donating an electron to the complex 3 and it is donating the electron to the complex 4. This is how it will be. Kindly make a note of it. This can also be expected in our exam. Now, so far we have completed how the electron being transmitted from in all these complexes, and what will be the function of this mobile carriers, and what will be the proton gradient, and what will be the whenever the electron has been flowed, what will be the energy is releasing, and the energy is helpful to pump the proton from the matrix region to the intermembrane space. Now, how the ATP is being produced, and the production of ATP, and that process we are saying with the means of this theory, that is chemiosmotic theory. And this theory was proposed by Peter Michel, and Peter Michel is the one proposed the chemiosmotic theory in 1961. And what will be the main concept? The electron flow create a proton gradient. The same thing, the electron has been flowed from one carrier to the next carrier. Whenever it is flowing, there is a release of energy, and that energy has been used to pump the proton, and as a result of this higher concentration of proton in the intermembrane space and lower concentration of proton in the matrix, this creates a proton gradient, and this gradient is the one storing the energy. I see here gradient storing the energy. The proton flows back into the matrix through the ATP synthase complex, that is the F0 F1 complex. The proton is bringing back from the intermembrane space to the matrix region, and this proton pump is one again driven for the ATP formation. See, and this ATP formation, here we are calling them as an oxidative phosphorylation. We have studied what is substrate level phosphorylation and oxidative phosphorylation, and this is the oxidative phosphorylation. Here the process of ATP formation using energy from the electron transfer in the sense. So what is the difference between substrate level phosphorylation and oxidative phosphorylation? Substrate level phosphorylation, directly the ADP, by accepting one phosphate, they are producing an ATP. This is what a direct synthesis, we are calling them as a substrate level phosphorylation. And in the oxidative phosphorylation, whenever the electron been transferring, some amount of energy is releasing, and that energy is used to produce ATP. Energy is used to produce energy, and this indirect use of energy from the proton transfer or electron transfer, we are saying that as a oxidative phosphorylation. And what will be the mechanism, how it is getting done? See here, the proton accumulate in the intermembrane space, we know that, and they move back to the membrane via the ATPase complex. We are saying that as a complex 5, ATP synthase, we know that, and this powers the ATP formation. How they are producing an ATP? ADP plus phosphate group, they are adding the by adding these two, we get ATP. So what is the ATP synthase structure? What will be the structure of the ATPs? We are having two main parts, just like I said before. We are having an F0 and F1 complex, and the F0 will be present in the membrane portion, and F1 will be present in the matrix side, and this F1 is the one involved crucially in the ATP synthesis. This F0 is the one involving in channeling all the protons. Now, See, ATP yield from the NADH. So we have studied complex 1 pathway and complex 2 pathway, that is NADH and FADH₂, and here 1, 3, 4 has been involved. These complexes has been involved. Here 2, 3, 4 complexes has been involved here. Okay, so now passes from the complex 1, 3, 4. So this is what from the ADP, NADH. NADH is giving the electrons to complex 1. From complex 1 it transferred to 3, and from 3 it has been transferred to 4. And more protons been pumped here. And we have studied how many ATP is being produced. 3 ATP. In the complex 1 pathway, in the classic value we are saying 3 ATP, and in the modern value we are saying 2.5 ATP. In the sense, what you should know, 1 NADP is equal to, we have said in the starting stage itself of cellular respiration, 1 ATP is equal to 3 ATP. But now we are saying 1 ATP, 1 NADH, they are producing approximately 2.5 ATP. So in yesterday's class I have explained you how 1 NADH is equal to, I have said that 1 NADH is equal to 3 ATP and 1 FADH₂ is equal to 2 ATP. It is approximately equal to. So in this class you come to know that how 1 NADH is equal to 3 or 2.5 ATP, and this is what an electron transport chain. So here where we have said by pumping the electrons they are producing an energy simply, and this transport of electrons we are saying that is an electron transport chain, right? Now from the NADH, so we have said that FADH₂, that is the complex 2, will not involving in any pumping of protons. So that's why we are saying fewer protons pumped, and the ATP produced will be 2 ATP and the modern value will be 1.5 ATP. So this is how it will be. So when compared to NADH and FADH, NADH will be producing more ATP. Now see here, this is very very important slide. So the question can be asked from here. See here, to glycolysis 280. 2 NADH, and 5 ATP will be yield at the end, which means 1 NADH is equal to approximately 2.5, and we are having 2 NADH in the glycolysis process, so we can say 2.5 by 2.5 plus 2.5 is equal to 5. So we are having 2 NADH in glycolysis, so as a result 5 ATP. And in pyruvate decarboxylation, again we produce 2 NADH, so 2 NADH again it is equal to 5 ATP. And Krebs cycle we are having 6 NADH, because for 1 acetyl CoA per turn we are having 3 NADH. Twice the 2 acetyl CoA produces how many? 6 NADH and 2 FADH. So 6 into 2. It's not like that. Yeah, 3 into 2.5 is equal to 7, and 7 plus 7 is equal to 15 approximately. See, let me explain you clearly. Give it. See here, we are having 3 NADH in what? Krebs cycle. Per 3 NADH is equal to 1 NADH is equal to 2.5 ATP. Then 3 NADH is equal to 7 ATP. Isn't it? And again 1 NADH, 3 ATP. No, I'm confusing. Yeah, wait. See here, 6 NADH. 1 NADH is equal to 3. So 6 NADH is equal to for 1 NADH is equal to 3 ATP, and in the Krebs cycle we are having 3 NADH. So 3 into 2.5 is equal to 7.5 ATP, and again twice the acetyl CoA, so we can say 6 NADH. So 3 already it is there, and 3 into 2.5 is equal to 7.5, so totally 6 NADP, so 50. So 15 ATP. So that's why here and FADH₂, one FADH₂ is equal to approximately 1.5. So totally 2 FADH₂, so 3 ATP will be produced. So finally, with the production of this NADH and FADH₂, in Krebs cycle we are having 18 ATP. So this is what we can expect in the exam. So totally in the means of this FADH and NADH, how many NADH? 10 NADH plus 2 FADH₂ will be produced in the cellular respiration. And as a result, we are having how many? 28 ATP. And besides, we are having ATP is also produced. So here we have calculated only with respect to this NADH and FADH₂. Now, what will be the role of oxygen in the electron transport chain? So this will be the final acceptor of electron. We have studied that by combining with the electron and proton, they are forming a water. The same thing. And without oxygen, if there will be no oxygen, the electron transport chain never happens. There will be the accumulation of NADH and FADH₂. And the Krebs cycle is also stops, ATP production ceases. If there will be no oxygen, all cellular respiration stages will be shut off. Now, see here, what are all the inhibitors which plays an important role in inhibiting different complexes? See, the complex 1 has been inhibited by rotenone amidal. This can also be explained. Whenever you are studying about this inhibitor, you have to be very careful. The question can be arise and it may tend to confuse you. So complex 3, in this complex 3 we are having an inhibitor, that is antimycin A. And in the complex 4 is cyanine carbon monoxide acid. So these are all the inhibitor which is inhibiting the complex 4. Now, so see here, very important, this NADH complex 1 is pumping 4 protons. Very important. This can also be expected. How many proton will be pumped by this complex 1? The complex 1 is pumping 4 protons and complex 2 is pumping no proton. We have studied about that. And complex 3 is pumping 4 protons and complex 4 is pumping 2 protons. So this is very important. So we are having 4 complexes by subtracting this ATP synthase, as these are all the complexes which are involving in pumping the protons. We are saying 4 complexes, that is complex 1, 2, 3 and 4. Complex 2 is not going to pump any protons, that is the succinate dehydrogenase. Complex 1 and 3, they are pumping 4 protons and complex 4 is pumping the 2 protons. This can also be expected here. And see here, the complex 1 will be inhibited by rotenone and 3 will be inhibited by antimycin and 4, this ATP synthase will be inhibited by oligomycin. Very important. That is, oligomycin is very important in question. So this will be expected. Now, uncouplers. What is mean by uncouplers? Uncouplers disrupt the proton gradient. See, uncouplers, which means inhibitors, effect the electron transport continues, but the ATP synthesis will be stopped. Electron energy released as heat. The energy will not be released. Energy is not used to produce an ATP. Instead, they released as heat. Examples: DNP, 2,4-dinitrophenol and thermogenin brown fat. So these are all called as the uncouplers. Again, it is also inhibiting the ATP synthase. Now, what are all the most important differences between this NADH and FADH₂? NADH is a complex 1, FADH₂ is a complex 2, and this proton pumping will be more in NADH and it will be less in the FADH₂. ATP will be higher in the NADH and lower in the FADH₂. This is a glimpse of it. And again the same thing. For your understanding, I will add another important picture. So that's all about this cellular respiration, and the entire biochemistry has been completed with this question. So we'll see the question. The electron transport chain in eukaryotic cell is located in electron transport chain. In case of eukaryotes, it will be located in which inner mitochondrial membrane. Very important. And which complex of the electron transport chain does not pump the protons across the membrane? And that will be the complex 2, that is succinate dehydrogenase. The final acceptor, who is the final acceptor? That will be the oxygen. That is oxygen accepts the electrons, the complex 4. This is the main important thing. And coenzyme Q functions as the mobile electron carrier. Not only coenzyme Q, the cytochrome C is also considered as a mobile carrier. And what about which complex is considered as which complex contains the cytochrome oxidase, that is complex 4. And the ATP synthase enzyme is also called as the complex 5. You know that chemiosmotic theory, that is Peter Michel. And how many ATP molecules are produced per NADH in modern calculations? That is, in modern calculations they are asking, that is 2.5 ATP. FADH is 1.5. And which of the following complex inhibits the complex 4 of the ETC, that is none other than the cyanide. And the main purpose of proton pumping in the electron transport chain is to main purpose of proton pumping in the electron transport chain is to create a proton gradient. So far we have completed 2 units, cell biology and biochemistry. We'll meet you with the next chapter in the next class. Thank you so much.